My name is Jorge Santillana, and today I will be presenting CRISPR-Cas9 mediated cervical cancer treatment targeting human papillomavirus E6. This is a basic overview of what the presentation will entail. 1. Starting with the background of what CRISPR and Cas9 is. 2. Would be the main question the researchers were trying to answer. 3. Methods they used to get these answers. 4 would be the results that they came up with, and 5 would be conclusion. So, what is CRISPR-Cas9? CRISPR-Cas9 induces double-stranded breaks at specific target DNA locations by the combined action of two key molecules that introduce a change, also known as a mutation, into the DNA. These are an enzyme called Cas9, which acts as a pair of molecular scissors that can cut two strands of DNA at a specific location in the genome so that bits of DNA can then be added or removed. The other key molecule would be an RNA guide, also known as a guide RNA. This consists of a small piece of pre-designed RNA sequence, which is usually about 20 bases long, located within a longer RNA scaffold. This guide RNA is designed to find and bind to a specific sequence in the DNA. The guide RNA has RNA bases that are complementary to those of the target DNA sequence in the genome. Scientists can then use a DNA repair machinery to introduce changes to one or more genes in the genome of a cell of interest. In this case would be the E6. CRISPR-Cas9 can be used to induce specific knockout of E6 expression in cervical cancer cells. However, application of such a strategy in clinical practice requires an effective vector that enables gene transfer into targeted cervical cancer cells. Cervical cancer. Cervical cancer is caused by high-risk human papillomavirus infection. Specifically, persistent infection of the basal epithelial cells of the cervix causes the integration of the HPV genome into the host chromosome. This leads to expression of the HPV E6 oncoprotein that inactivates the tumor suppressor gene P53, resulting in tumor formation. Furthermore, the expression of E6 is limited to cervical lesions, with no expression in healthy tissue, including the cervix. So suppressing the expression of E6 may lead to the potential treatment of cervical cancer. Therefore, specific targeting of E6 expression is likely a safe strategy that would spare normal tissue from additional harm. The main question of this research was to find an approach on cervical cancer without any harm. By combining cervical cancer cell lines engineered to constitutively express Cas9 and an adeno-associated virus vector carrying a single guide RNA targeting E6, the present study sought to investigate the effects of this novel therapeutic approach on cervical cancer HPV E6 oncoprotein, which promotes the degradation of the host tumor suppressor gene P53, which and then leads to development of tumors. Therapeutic strategies that specifically target E6 may be highly effective and safe. So, basic methods they use in this experiment would be cell culture, RT-qPCR, and Western blot analysis. For the cell culture, they got 18 positive human cervical cancer cell lines. The three that they use were HeLa, HES2, and SKG1, and human immortal cell line 293. These cells were cultured in Dulbeckel's modified Eagle medium, F12 supplemented with 10% inactivated fetal bovine serum and 1% penicillin streptomycin at 37 degrees Celsius under 5% CO2. Tumor cells were seeded on the PCR on 6 well plates at 5 times 10 to the 4th cell walls, incubated for 24 hours, and transduced with either AAV SGE6 or AAV SGNC, which is known as a negative control, which would have nothing on it. Cells were collected by trypsin 48 hours later, and DNA was extracted. qPCR was performed according to the protocol using the thermal cycler DICE real-time system. The PCR was carried out using 40 cycles of heating at 95 degrees Celsius for 15 seconds, 58 degrees Celsius for 15 seconds, and 72 degrees Celsius for 20 seconds. In methods in vivo. So they used 6 to 7 week old BALB/c nude mice, which were used in the experiment. They had a total of 8 This rat lacks a thymus and is unable to produce T cells and is therefore immunodeficient. SKG1 was selected among the 3 cervical cancer cell lines as it has been reported to induce tumors in nude mice. A total of 5 times 10 to the 6 Cas9 expressing SKG cells were injected under the dorsal skin of nude mice. Simultaneously, 2 times 10 to the 12 were injected once in the same location. Changes in the body weight were measured and gross observation of the injection site was performed at an experimental endpoint. DNA was extracted from tumors in both groups using a DNA mini-kit, and the E6 mutation rate was measured by the qPCR as described above. Multiple tumors were not observed in the present study. Methods for the in vitro cell growth. Tumor cell lines, they use the Cas9 gene was transfected into 3 high-risk HPV positive cervical cancer cell lines, the HELA, HCS2, and SKG1, to establish cell lines that constitutively expressed Cas9. In order to confirm that the Cas9 is introduced, we performed Western blots for transfected cells using the anti-HA antibody. Cas9 protein was expressed only in those cells that received the Cas9 gene. We can see this in figure A. Cas9 expressing cervical cancer cell lines HELA, HCS2, and SKG1 were established. In order to assess the effect of Cas9 on cell growth, we compared the growth of wild-type and Cas9 expressing cervical cancer cell lines in vitro, as shown in figures B through D. There was no significant difference in cell growth between wild-type and Cas9 expressing cells, indicating that Cas9 expression had no impact on cell growth in vitro. Detection of E6 gene mutations. The E6 gene was sequenced by Sanger sequencing for HELA cells 48 hours after vector SGE6 transduction. Several base insertions and deletions were identified mostly around the targeted region, as seen in figure A, and this would be the green and red area that is on the base pairs up top. As you can see, there is a 13 base pair deletion on the second line, then a 20 base pair deletion on the third line, and then the 2 insertions on the last 3. Products cleaved by the T7E1 were detected only in cells transduced with the vector SGE6. Figures B through D show us that gene mutations were introduced into the E6 gene by E6 targeting CRISPR-Cas9. qPCR was then performed using mutated plasmid vectors as templates and demonstrated a significant increase in mutation rates. This is seen in figure E. In addition to that, mutation rates increased with an increasing proportion of mutated plasmid vectors, and this rate was equivalent to the proportion of the mutated vectors in the plasmid mixture. The rate of E6 mutation was measured using the same method. Mutation rates were estimated to be 82% for HELA, 77% for HCS2, and 87% for SKG1. This is just a continuation of the previous slide showing the mutation rates for the last 2 cancer cell lines. Results in vitro cell growth. In figure A, the expression of E6 was examined and using Western blotting found decreased expression of E6 in all 3 cervical cancer cell lines. E6 expression was detected in untransduced cells or cells transduced with AAV SGNC. These cells did not express P53. On the other hand, cells transduced with vector SGE6 had significantly decreased expression of E6 and significantly increased expression of P53. These results indicated that E6 was knocked out effectively by CRISPR-Cas9 targeting of E6, consequently increasing the expression of P53 at the protein level. In high-risk HPV positive cervical cancer, P53 has no mutation and is normally degraded by resulting inhibition of apoptosis but in this case, the suppression of E6 expression by Cas9 and vector SGE6 led to alteration of this pathway, resulting in the induction of apoptosis. Figures B and C show us that E6 targeting CRISPR-Cas9 induced apoptosis in tumor cells in vitro. Apoptotic cells are stained with immunofluorescence staining, and the number of cells was counted in a high-power field. This is why we can observe a high number of cells in vector E6 as compared to the negative control, since no apoptosis is occurring. For figure D, the WST1 assay was performed to measure the number of viable cells 72 hours after vector SGE6 transduction. Vector E6 suppressed tumor growth with increase in viral vector particles in all three Cas9 expressing cell lines. Results in vivo. In vivo experiments, tumor growth was suppressed significantly in mice injected with the vector SGE6 as compared with those injected with the negative control. As you can see in figures A, which is on the top left hand side, you can clearly see differences between the tumor volume after the halfway mark between days 14 and 21, which is where the tumors of the negative control start to grow at a very fast rate compared to those of E6. Figure B is more towards the experimental endpoint of 42 days after the injection, and the tumors in the vector SGE6 group were significantly smaller than those in the vector SGNC group, as you can clearly see the difference in size between the two. In figures C and D, you have the tumor weight, and the tumor weight of the negative control is way heavier than the vector E6. As you can see in the picture, you wonder why, because it did not stop tumor cell growth. Throughout this research, they were able to provide a report that the CRISPR-Cas9 approach targeting high-risk HPV-E6 may be a highly selectable and effective therapeutic strategy for cervical cancer. In order to take this into a clinical approach, it may be tested with patients with precancerous lesions as well as early-stage invasive cancers. Currently, surgical approaches such as cervical conization and a full hysterectomy are only options to treat patients in early cancer development. The effects of CRISPR-Cas9 are localized, therefore are unlikely for an advanced cervical cancer patient with widespread invasion and metastasis, but it can shrink down bulky tumors and may be used as an alternative to neoadjuvant chemotherapy.